In this chapter, we're going to learn how the rates of chemical reactions in your body, how those rates are controlled, which means we're going to learn how chemical pathways, how the rates of different chemical pathways that could be catabolic or anabolic, how those pathways, the rates can increase or decrease. As we mentioned before in our first chapter, every chemical reaction in your body, there's a catalyst, and the catalysts in your body are enzymes, which are proteins. We're going to understand by the time we're finished with this chapter how those chemical reactions are controlled, and it's related to the function, the activity of the enzymes. Certain enzymes in your chemical pathways, some of those enzymes are controlled, meaning those proteins can work better under some environmental conditions or work worse under another type of environmental condition. If you remember from our discussion in the first chapter, I wanted you to think of concentration of a molecule in your body as a lake level. And there's rivers flowing from that lake, and there's rivers flowing into that lake. The rivers are like the chemical pathways. And also I want you to remember that a major strategy for all organisms is to keep concentrations of molecules constant or relatively constant in your body. So here's our lake level, and there's some sort of concentration, and that concentration is based upon the lake level. Remember, brackets like this means concentration. There can be flow out, and there can be flow in. If the flow out of the water and the flow in of the water, if it's the same rate, then that lake level is constant, and concentration is constant. Think of flow out, and there can be multiple rivers flowing out. That's usage of some molecule. Flow in would be production of whatever molecules in that lake. And in these rivers, these chemical pathways, there can be dams, so that's a dam. And dams like on the Spokane River can be controlled. They can be opened and closed. That dam could be an enzyme that's part of a chemical reaction that's part of this pathway. And that enzyme, it could work better or work worse. It's like opening and closing the dam. So you can control the water flowing out. There could be multiple dams along the river. There could be both outlets of rivers. There could be a dam. There can also be dams that are coming in to that river so that whatever concentration is in there, but there could be a dam in there. We'll learn how these dams are controlled. And that control, we're controlling some protein, and that protein is changing shape, and its activity and its function is changing. The chemicals that are around that protein, and that protein could be an enzyme or it could be a transport molecule into a cell, but the chemicals around it are controlling how well that protein is working. A lot of the information, well, all the information that we learned in the previous chapter dealing with proteins and understanding how their shape can change, we'll see those same concepts in this chapter. In fact, when you go and look at the practice problems, there will be a lot of similarly worded problems because we're dealing with enzymes in this chapter, which are proteins. So as far as this lake level, when you have some sort of molecule and the concentration is constant, things can change. Like, for example, ATP concentration in your muscle cell. Well, if you start exercising, its usage is going to increase. ATP is a reactant for muscle movement. And for any molecule in your body, if its usage increases and production is constant, the concentration of that molecule is going to change. If production is constant, you should be able to say what happens to concentration. It either increases or decreases. Why don't you pause the video and unpause the video when you're ready to check your answer. The concentration would decrease. So this would be analogous to opening up this dam, and all of a sudden that flow out increases, but the flow in was constant. Then that lake level would decrease. Concentration would decrease. And if the concentration did decrease, then your body's going to do something to increase the production, to increase the flow of water back into that lake to keep it constant. This would be like on Lake Coeur d'Alene. What if you opened up the dam? It would be nice if you could say to nature, rain some more, produce more water to flow in because you opened up the dam for the flow out. Your body can do it. Can't do that in nature. We're still working on it, but you're pretty amazing that you have that ability to control your chemical pathways. Okay, just the opposite. What if production increased and usage was constant? The concentration, that lake level, would change. So all of a sudden you have a big flow in, the flow out was constant, that lake level would increase. So concentration would increase. This would be like we had the storm of the century, had tens of inches of rain fall in Wallace or Kellogg, and there was a lot of water that flowed into Lake Coeur d'Alene. These dams, if they were kept at the same level, the lake level would increase. What would a Vista do? Well, a Vista would open up the dam to let more flow out. That's the control. And again, your body, if there was some chemical pathway and the flow in increased for some concentration, these enzymes or proteins downstream that control the pathway out would work better. And we'll see how that works as we look at the pathways more specifically in upcoming chapters. But again, it's a major strategy for most molecules in your body that aren't waste molecules to keep those concentrations constant, to keep that lake level constant. It can vary up and down a little bit, but it's pretty close to being within that average concentration throughout the day. On the next two slides, we're going to look at two separate variables that affect the rate of a pathway. The first one is concentration. If the concentration of a reactant that's part of a pathway, if it increases, the rate of that pathway, the rate of the reactions in those pathways will increase. If concentrations of reactants, if they decrease, then the rate of the reaction, the rate of the pathway will decrease. So let's put a marking around that. As reactant concentration increases, rate of reactions will increase, or the rate of the pathway will increase, and vice versa if it decreased. So let's give an example of a pathway that's in your body. The name of this pathway is called oxidation phosphorylation. And in the carbohydrate metabolism chapter, we'll learn more about that. But here's the net reaction. This is the pathway that, it's the only pathway in your body where oxygen is a reactant. So there's our reduced coenzyme, NADH. It's carrier of 2 electrons, carrier of chemical energy, reacts with a hydronium ion, also abbreviation hydrogen cation, to form water and NAD+. You don't need to memorize this pathway. In fact, this pathway is given in your information packet, and shown down here is oxidation phosphorylation, and also, well, I did it twice. This pathway can begin with either reactant, NADH or FADH2. And we'll learn more about that in an upcoming chapter. We're just using this pathway as an example in this chapter. This is a net reaction, but if one of the reactants, if one of the concentrations change, the rate of the pathway is going to change. And let's assume that oxygen's concentration changed. And let's assume that it decreased. And you could do this just by holding your breath. But let's say oxygen concentration decreased. The rate of oxidation phosphorylation, this pathway, would change. Why don't you go ahead and pause the video and fill in that blank. Unpause the video when you're ready to check your answer. If one of the reactants' concentrations, if it decreased, then the rate of the pathway would decrease. And down here is kind of a picture diagram of this process. So we have NADH, and it's producing water, so NADH producing water. And we have oxygen, O2, as a reactant, and hydrogen ion, H+, as a reactant. If oxygen's concentration decreases, there's less oxygen to collide with NADH, and the production of water would decrease. Assuming that the flow in is constant, so that river flow in into NADH is constant, so assuming the production of NADH is constant, its concentration, its level here would change. Why don't you go ahead and pause the video and fill in that blank. Unpause the video when you're ready to check your answer. That lake level of NADH would increase. When oxygen's concentration decreases, this river out is slowing down. It's like closing the dam on the Spokane River. That flow out decreases, the flow in is constant. That would increase the concentration of NADH. One thing to note, oxygen is a reactant in oxidation phosphorylation and only oxidation phosphorylation. So changing its concentration only affects this pathway. It doesn't affect this inlet pathway to produce NADH. Meaning for this particular pathway, only the outlet is affected by oxygen, not the inlet. Eventually, there's some other indirect interactions that affect the inlet and the production of NADH, but not initially. And this is true for most pathways. Either the inlet or outlet is affected, not both. One remains constant, the other either increases or decreases. Okay, let's look at another aspect of this pathway. NADH is a reactant for oxidation phosphorylation. It's also a reactant to produce lactic acid, which is also called lactate. NADH is a product in many pathways, and it's a reactant in many pathways in your body. Shown on this page are two pathways in which it's a reactant. This pathway from NADH to lactic acid is also shown in your information packet. This is a pathway that we'll learn about in the carbohydrate metabolism chapter, so you don't need to know anything about this reaction quite yet, other than we're using it as an example reaction. NADH reacts with pyruvate to form lactic acid, also called lactate. But let's assume, and let's make another problem for you to fill in the blanks. Let's assume NADH's concentration is increasing. So just following from what we just said in our previous example, this lake level is increasing. If cellular concentration of NADH increases, then the rate of producing lactic acid would, now I want you to fill in that blank. So the rate of this reaction would change, and the concentration of lactic acid would either increase or decrease, assuming that the usage of lactic acid is constant. Go ahead and pause the video, fill in those two blanks, unpause the video when you're ready to check your answer. Okay, so as NADH's concentration increases, the rate of that pathway to produce lactic acid would increase because NADH is a reactant in that pathway. So increase, which means you'd produce more lactic acid, assuming that the usage of lactic acid is constant, then the concentration of lactic acid would increase. This, hopefully you're seeing, is showing the beginning complexity of your chemical pathways in your body. Oxygen, it's not used to produce lactic acid, or it's not used in the production or usage of lactic acid. It's only used in this pathway, but indirectly, oxygen affects the production of lactic acid because oxygen is a reactant with NADH, and oxygen can affect NADH's concentration. NADH is also a reactant in the production of lactic acid. NADH is the molecule that links the two pathways together. So as oxygen's concentration changes, it indirectly affects lactic acid concentration. And this is why some of you have heard when you exercise intensely, your lactic acid production, it increases. On the previous page, we learned how concentrations of reactants can affect the rate of the reaction. On this page, we're going to learn about the concept of inhibitors and activators. These are chemicals that don't chemically react in the pathway, but their concentrations affect the rate of the pathway. Activators and inhibitors, these are chemicals that are in your cellular fluids or in your blood, and these are chemicals that attach to the proteins and either make the proteins work better or work worse. An activator binds to a protein, and that protein's shape, its conformation changes, and the protein works better. We gave an example of this in our previous protein chapter when we looked at hemoglobin and the shape change effect caused by carbon dioxide. As the concentration of carbon dioxide increased, the conformation of hemoglobin changed, its shape changed, such that hemoglobin worked better at higher carbon dioxide concentrations. In this chapter, we're talking about enzymes and not a protein that carries oxygen, but nonetheless, an activator is an activator. An activator for certain enzymes make the enzymes work better. Inhibitors, they make it so that those enzymes don't work as well. There's symbols for inhibitors and activators, and for an inhibitor, having a circle with a slash through it, that means that chemical is an inhibitor. For an activator, if you see a circle with an up arrow, that's an activator, or a square with an up arrow, that's an activator. If we go to our information packet, there's listings of different inhibitors and activators for different enzymes. Here in the information packet is a table for glycolysis, and again, we'll understand this chemical pathway more later on as we get into the carbohydrate chapter. But here's those 10 reactions. Here's the name of the enzyme for reaction number 1, and we see glucose 6-phosphate with a circle. It's an inhibitor. You don't have to memorize inhibitors and activators for enzymes. If we look at enzyme number 3 for reaction number 3 of the reaction, there's 2 inhibitors for that particular enzyme and 1 activator. Notice for the enzymes in this pathway, the ones that are shown in this pathway, 3 out of the 7 enzymes are controlled, have activators and inhibitors. You don't have to control every enzyme that's part of the pathway. It's like the Spokane River going through, the river going through the Spokane area. There's like 9 dams. You just need to close 1 dam to affect the flow of the water throughout the whole Spokane City area. Those activators and inhibitors that I just showed you in glycolysis, those are chemicals that are part of your metabolic pathway. Those are chemicals that your body produces and uses up. The chemicals that are part of your metabolic pathways, they control the enzymes that are producing or using up those chemicals. So inhibitors and activators are chemicals that are part of your metabolic pathways. We'll give an example of ATP on the next page, and you're going to decide if ATP is an inhibitor or an activator. But let's first learn a few more basic concepts about these chemicals. Let's first start with an activator. An activator, it binds to the protein, to an enzyme, and there's a specific location for that activator to bind. As it binds, it causes a conformational change, a shape change, and the enzyme works better. The effect of the activator on the enzyme depends upon the concentration of the activator. And the reason why it's concentration-based, the higher the concentration of the activator, the more likely it will collide with the enzyme and attach and cause a conformational change in the enzyme. So we're going to describe the effect of the activator based upon its concentration. So we're going to start out a statement as an activator's concentration increases, and then we'll have you fill in the rest of the statement. But remember what an enzyme is doing. The enzyme is weakening the bonds of the reactants. So if the enzyme is working better because of the activator, it's doing a better job at weakening the bonds. So as an activator's concentration increases, the bonds of the reactants are weaker. The activation energy, remember what activation energy is, it's the energy required to break a bond. So if the bonds are weaker, then the activation energy changes. So I want you to fill in these 2 blanks. So as the activator concentration increases, the activation energy, the E sub A, either increases or decreases. And the rate of the reaction, remember, depends upon the activation energy. So the rate of the reaction will either increase or decrease as the activator concentration increases. Pause the video, fill in those 2 blanks. Unpause the video when you're ready to check your answer. So if the bonds are weaker because there's more activator attached to the enzyme, the activation energy is less. If there's a lower activation energy, there's more molecules in there that have enough energy to break the bonds, and you increase the rate of the reaction. Okay, the opposite happens as the activators, as its concentration decreases. The activation energy would increase, and the rate of the reaction would decrease. If there's less activator in there, that means the enzyme's not working as well, which means the bond strength are increased, which means the activation energy increases and the rate of the reaction would decrease. Inhibitor molecules, their interaction with the enzyme, with the protein, it's the opposite. When the inhibitor attaches, there's a conformational change in the protein in the enzyme, and the enzyme doesn't work as well. So as the inhibitor concentration increases, we can make a statement just like those two. We can say something about activation energy and the rate of the reaction. So as the inhibitor molecule increases, there's more of it attached to the enzyme's not working as well, so the bond strength increases, which means activation energy increases, which means there's less molecules that have enough kinetic energy to break the bonds. If the inhibitor concentration decreases, then there's less of a chance that the inhibitor would be attached to the enzyme, making the enzyme not work as well, which means the enzyme's actually working better with less inhibitor attached. So the bonds are weaker and the activation energy would decrease, and the rate of the reaction would increase as the inhibitor concentration decreases. So this may take flashcards in the beginning to get these four scenarios straightened out in your mind, but you do need to know that, and you'll see practice problems in your study guide. But let me give you one right now and let you predict what's going to happen. So I'm going to give you a chemical reaction, and I'm going to tell you whether there's a molecule that's an inhibitor or activator for it. So we'll see this chemical pathway later, but when fatty acids are catabolized, they're broken down into two carbon segments, and we form a bunch of acetyl coenzyme A's. This is a pathway, it's a multi-step process, but NADH is an inhibitor to this pathway. So let's assume that NADH's concentration increased. If NADH concentration increased, so that's given to you, what would happen to this pathway? The rate of the pathway would, I want you to fill in that blank. So pause the video. Would that pathway increase or decrease if the inhibitor concentration increased? Unpause the video when you're ready to check your answer. If there's more inhibitor in there, those enzymes that are part of that pathway aren't working as well, so the rate would decrease. So NADH has an effect on the fatty acid catabolism. NADH is not a reactant or a product. It's a molecule produced in other parts of your pathway, but it does have an effect on a pathway that's part of that cell. This is what adds complexity to your metabolism. Chemicals that aren't part of that pathway, they're not reactants or products, have an effect because they interact with enzymes that are part of that pathway. So if we looked at our previous page, there's NADH, it's part of those two pathways and others, and its effect can have an effect on another pathway. Oh, by the way, NADH concentration increasing, how did that happen? That could happen by oxygen concentration changing. As oxygen concentration decreases, we saw that NADH's concentration increased. And that increase in NADH had an effect on lactic acid. It would also have an effect on the breakdown of fatty acids. So indirectly, that change in oxygen concentration, even though it's not chemically reacting or part of that pathway, it's not an inhibitor or an activator, indirectly, oxygen, we've just seen, affects two different pathways. Enzymes are our biological catalysts, and virtually every enzyme that's in your body is a protein. And every different type of chemical reaction, there's a different protein, a different enzyme that has been synthesized for it. The control of these proteins, of these enzymes, is pretty complex. And I'm going to draw out a schematic of some pathways that we're going to be looking at. And I want you to be able to predict if a molecule is an inhibitor or an activator. So let's start out with glucose. And I'm going to draw arrows for those 10 reactions of glycolysis. So 4, 5, 6, 7, 8, 9, 10. The end molecule of glycolysis is pyruvate. Pyruvate, as we'll learn a little bit later, can be broken down, catabolized, and there's more than 3 arrows, but we'll just kind of end at that, and produce NADHs. We'll also learn later that the chemical energy that's in NADH can be used to make a lot of ATP molecules. Pyruvate can also be used to form lactic acid, so lactate. The ATP that's been produced, ATP is a reactant in hundreds of reactions. One reaction, as we've mentioned a few times, is muscle movement. So ATP can be used for muscle movement. It can also be used to make other molecules like proteins. So ATP is a reactant for the building of big proteins from amino acids. Some other molecules that we're going to be looking at for synthesis, you have the ability to synthesize glucose in your liver, and you can make glucose from amino acids. We'll also learn a little bit later that the lactic acid that's produced in your cells can be secreted into your blood and taken up by your liver, and glucose can be synthesized from the lactic acid. You also learned a little bit earlier in a previous chapter in 122 that glycogen is made from glucose. It's a storage polymer for glucose, and it can be broken down and reform glucose. We'll also learn that the fat that's in your body, well, that fat can be broken down, if you remember from the first chapter, into fatty acids and glycerol. That glycerol that forms can be converted into a molecule that's part of the pathway that breaks glucose down into pyruvate. So these are streams. If you want to think of this as our main river, here are streams that are flowing into that river. Okay, what I'm going to have you do next is to try and predict if a certain molecule is an inhibitor or an activator. It's ATP. And ATP is an inhibitor or activator for reaction number 3 that's part of this glycolysis pathway. And I'm going to have you try and predict, based upon logic, is it an inhibitor or an activator? Okay, so what I wrote there, enzyme number 3 is controlled by ATP concentration. It's an inhibitor or an activator. So as we go through this process, remember a strategy in your body is to try and keep the concentration of molecules constant, to try and keep that lake level of ATP constant. And ATP, it's downstream, it's produced, the chemical energy in glucose can be used to produce ATP. So let's assume that you started to exercise, and ATP, it's used up when you exercise, when you have muscle movement. So as you start to exercise, the usage of ATP increases. So let me read what I just wrote. As you start to exercise, the usage of ATP, there's the up arrow, increases. There's no reason why the production of ATP would change when you exercise. None in the beginning. So assuming that ATP production is constant, the concentration of ATP would decrease in your cellular fluids. This is something you actually should know for the rest of the quarter. As you exercise, ATP is used up and its concentration decreases. We'll see that in a lot of problems throughout the quarter. So if this usage increases, that would cause that lake level, the concentration of ATP, to decrease. You'd hope that the dams that are here would open up, which means you would hope that this enzyme would work better as ATP's concentration decreases. What does that mean? Does that mean ATP is an inhibitor or an activator of enzyme number 3? Why don't you go ahead and pause the video and give a good educated guess at whether ATP would be an inhibitor or an activator of that enzyme. Unpause the video when you're ready to check your answer. The enzyme is working better when the ATP concentration decreases. That must mean that it's an inhibitor. Okay, some of you at this point may be worrying that I have to be able to predict inhibitors or activators or memorize them. Well, you don't. In fact, here in our information packet, you're shown ATP being an inhibitor for enzyme number 3 for that part of glycolysis. As always, look at the study guide with the practice problem as a guide for the questions that are going to be on the exam. It gets you directed on how you have to learn the material. I certainly don't expect you to memorize all these inhibitors and activators. But hopefully, as we do present these pathways in more detail in upcoming chapters, it makes sense when we see that a particular molecule is an inhibitor or is an activator. As mentioned earlier, enzymes are proteins. They're huge molecules. Somewhere on the protein is where the catalytic process occurs. This place where it occurs on the enzyme is called the active site. There's at least one active site per enzyme. A lot of enzymes have multiple active sites. It's kind of like hemoglobin, where there's 4 heme molecules, 4 binding sites for oxygen on hemoglobin. Enzymes can have multiple active sites. In our diagram over here, this is supposed to be our active site. This shape of this active site, it's complementary to the reactant. In biochemistry, the reactant is called the substrate. So this substrate, it must fit into that active site. We'll learn on the next few pages that a substrate, a reactant, is very specific to an active site. But definitely one thing that needs to occur is that that substrate needs to be able to fit into the active site for a catalytic process to occur. When an enzyme interacts with a substrate, there's different things that it can do to increase the rate of the reaction. But what's definitely true about the net result of an enzyme's interaction with the reactant is that the activation energy is lower and the enzyme is not consumed in the process. How it does this, there's different mechanisms. One thing that an enzyme can do is that it can weaken the reactant bonds. So what I wrote, enzymes lower the activation energy and is not consumed in the process. And here's number 1, by weakening the reactant bonds. How it weakens the bonds, we'll show an example of that a little bit later. Number 2, and we're going to see this number 2 mechanism on the upcoming slides as we flip through the slides, is that it chemically reacts with the substrate. So there's a different chemical mechanism with lower activation energies. So what I listed here for number 2, chemically reacts with substrate. And again, we'll see that example on the next page. There's a few theories that are listed on this slide that show how an enzyme interacts with a substrate. One of them is called the lock and key theory, and the other is the induced fit model. Both work in different circumstances for different enzymes. The lock and key, if this is the substrate, then that active site, its shape, it has a shape that's very complementary to that substrate, and it fits in, so lock and key. The induced fit model is that the substrate fits partially into the active site, and then the active site changes shape to fit it completely. Both theories actually work, and different enzymes, depending upon which enzyme they are, some fit the lock and key theory and some fit the induced fit model. On the next few slides, we're going to go through the general process of an enzyme catalyzing a reaction. In this particular process, the enzyme is going to chemically react. After we're done going through those slides, you should be able to make a list of what needs to occur between an enzyme and a reactant, such that that reactant is very specific to the active site on that enzyme. This list, I guarantee you, you'll have to be able to make this list on the upcoming exam. So in this particular example, we're going to take some sort of generic polypeptide, and in this reaction, that polypeptide, that peptide bond is going to be broken, and we're going to make two smaller polypeptides. If you remember, to break that amide functional group, that peptide bond, all you need is water. This reaction can occur with or without an enzyme. But the net reaction when we're done will be a polypeptide plus H2O, forming two smaller polypeptides, which I'll call P1 plus P2. And there will be some enzyme that's involved in the process. The enzyme in this particular example is going to chemically react, but when all is said and done with the process, it's going to be reformed into its original state. So if it makes a bond in the beginning, that bond needs to break in the very end. Okay, so let's give our examples. So on this page, here's our enzyme, which we'll classify as E, so E for enzyme. Here's our active site, and in this active site are R groups that are actually going to be involved in the process. Our substrate we'll call S, so S for substrate. So what's happening here is the enzyme collides with the substrate S to form an enzyme substrate. When this collision occurs, that substrate will collide with the active site. In order for a catalytic process to occur, the substrate needs to stay in there. When you first have this collision, it stays in there because of attractions, not making the bond originally, but because of attractions. So there's intermolecular forces that hold the substrate and enzyme together. So this ES, those two, it's called a complex, and a complex is something that's held together by intermolecular forces. So what I wrote there, complex that is held together by intermolecular forces. That's our E sub S. So hopefully you're starting to see the list that we have to make. So that substrate, it needs to be able to fit into the active site. There's a lot of chemicals that can fit into an active site. Water could fit into any active site in any enzyme in your body. But that substrate, it also needs to stay in there, so there has to be some sort of attraction. In our particular example, what's being attracted there is an R group that's hydrophobic. So maybe there's a hydrophobic pocket in there. Lots of R groups that you can have some dispersion forces for attractions. Next is step 2 in our general process. So in step 2 is where the chemical reaction occurs and a bond is formed between the enzyme and its active site and the amide functional group that's going to be broken in the polypeptide. And what happens is the lone pair, one of the lone pairs that's on the oxygen in this alcohol group in the active site, it makes a bond with that carbon that's part of the amide group. So the enzyme substrate complex, when that bond forms, that's a chemical reaction and we form something else. And we'll say it forms the enzyme substrate, and we'll call it number 1 because now there's a chemical bond between the two. Note in this process, in order for there to be a chemical reaction, these lone pairs have to be really close to that carbon. If this being the OH, the alcohol group, was shifted one way or the other, these two functional groups weren't aligned. So the functional group that's reacting on the substrate and the functional group that's in the active site, if they're not close to each other, and close means much less than a nanometer, you're not going to have a chemical reaction. So those functional groups have to be in close proximity. I bet that's going to be part of our list that we're going to make in a little bit. So that substrate has to fit in the active site. It needs to stay in that active site. And the functional groups that are interacting, they have to be in close proximity. Okay, so shown here is that chemical bond that was formed on the previous page. And so here's our enzyme substrate number 1. And I'm going backwards a slide. When this process occurred, I didn't really mention, but this H that's on the hydroxy group, on the alcohol group, this bond breaks, and that H makes a bond with the lone pair that's on the amine group on the other R group that's part of the active site. Next in the process, we see that this bond, that peptide bond, breaks in our amide group. And why would that break when this OC bond forms? Well, these electrons, you could explain it based upon electronegativity, and don't worry, I'm not going to make you explain it on the exam, but where are electrons more attracted to, oxygen or nitrogen? Well, they're more attracted to oxygen, and that's why that CN bond breaks and not a CO bond. But nonetheless, we get that bond that breaks, and that's a polypeptide. So we had our enzyme substrate number 1. It chemically reacts, that peptide bond is broken, and we form enzyme with only partially some of the substrate remaining, so we'll call S2, plus a polypeptide. And this polypeptide that formed, it's not attracted to the active site, so eventually diffuses away. On the next page, we'll finish this catalytic process. At this point, we still have part of that polypeptide attached to the active site. We haven't reformed the original active site. That needs to occur, and we haven't seen water react yet. The net result of a catalytic process is the same with or without the catalyst. So our net reaction, we need to have water that's part of the reaction. So here's our enzyme substrate number 2, and here's water that's coming in. The lone pairs on the water make a bond with that carbon. When that bond forms, it causes this bond to break. One of the OH bonds breaks, and we reform the alcohol group on the serine R group. When that O bonds, that OH bond forms, we make the carboxylic acid. The peptide, this peptide that's formed here, it eventually diffuses away. Remember, it's attached, but it's only attached via intermolecular forces, not a covalent bond that's super strong. So eventually that polypeptide can diffuse away. So our last chemical reaction, we had our enzyme substrate number 2 plus water forms the original enzyme that can be used over and over again, plus the second polypeptide strand. And if we did a net reaction, the enzyme is used up, but then it's reformed, so it's not part of the net reaction. We took a polypeptide and we made 2 smaller polypeptide chains, and we used up a water molecule. So with or without an enzyme, it's still the same net process. The water collides with a peptide bond, and we form 2 polypeptide strands. Chemical reactions can happen with or without the enzyme, meaning this reaction can happen with just water and the polypeptide, or it can happen with the polypeptide, water, and the enzyme. For it to occur with an enzyme, remember enzymes are very specific for certain types of reactants. There's a listing of 3 things that need to occur in order for an enzyme to catalyze a substrate. There are 3 things, and I expect you to be able to list these 3 things on the exam. We've already mentioned these. The first one is the substrate has to be able to fit into the active site. But that's just one of 3. There's lots of chemicals that could, in your body, that could fit into a polypeptide active site. Another thing that we mentioned earlier is that the substrate has to be attracted to the active site. And the reason why this is a prereq is the fact that the substrate, it has to stay in there so a catalytic process can occur. But there's lots of hydrophobic molecules that are in your body that can fit. There's lots of molecules that can hydrogen bond and maybe stick to an active site. The third prereq, and is just as important as the first 2, the functional groups on the substrate and on the active site that are part of the catalytic process, they need to be aligned. They need to be in close proximity. And when we say close proximity, less than a nanometer, maybe like 0.1 nanometer. So these are the three things that need to occur in order for an enzyme to catalyze a reaction. And again, I expect you to be able to list these three. It doesn't have to be word for word. If you want to use a different word besides proximity or aligned, as long as it makes sense, you can use your English verbiage. As mentioned earlier, the catalysts, the enzymes in your body, are super good at increasing the rates of reactions. And typical catalysts can increase the rates from 1 million to 10 million times compared to uncatalyzed reactions. If you just relied on the reactants reacting, it wouldn't be who you are. And a good example is the conversion of carbon dioxide, which is a waste molecule in your body, into carbonic acid. This is the first reaction that happens to carbon dioxide when it's produced in every cell in your body. In the form of carbonic acid, in that form, that's the form that flows from your cells to your blood and eventually to your lungs. You need this reaction to get rid of carbon dioxide. Carbon dioxide is very insoluble in water. When it's converted to carbonic acid, that's the form that's quite soluble in water and could be eventually excreted by your lungs. Without the enzyme carbonic anhydrase, this reaction would be about 10 million times slower in your cell. What does that mean for you? Well, if you didn't have this enzyme catalyzing this reaction, it would be really hard to get rid of the carbon dioxide. Your metabolism is based upon how much carbon dioxide you produce. The higher metabolism, the more fuel molecules you catabolize, the more carbon dioxide that's being produced. If you didn't have this enzyme, your metabolism, the number of calories you produce each day, would have to be about 10 million times lower, which means you wouldn't really move throughout the day because you couldn't produce that many calories. Shown down here is a picture of the carbonic anhydrase, and I don't expect you to have any of this memorized. It's just for illustration purposes only. Blown up in this area, this is where the catalytic process occurs, and maybe I'll do it right down there. Here it is blown up. At this active site, it's more than just the R groups. There's some histidine molecules, and don't worry if you don't know what a histidine is. But what's interesting, you also have hydroxide and a zinc ion. There's a metal zinc ion that's part of this catalytic process. Getting rid of carbon dioxide is important to you. Don't be low in your zinc ions. One thing to note about enzymes, when we've talked about it, we've kind of thought about the forward direction, the reactants forming the products. Enzymes can catalyze the forward reaction and the reverse reaction. And we can illustrate this by drawing a reaction diagram. So here's a reaction diagram. This is an exothermic reaction and has an activation energy. So here's CO2 and water and carbonic acid down here. And this is the reaction diagram without a catalyst. If you added a catalyst like carbonic anhydrase, you still start with the same reactants and you still end with the same product, so we're still at the same potential energy, but the activation energy is lower. And it's lower in the forward direction, but it's also lower in the reverse direction if we go in that direction. Chemical reactions are reversible. Enzymes lower the activation energy in the forward and reverse direction. This is pretty important because we'll see this later on. We have pathways where we break down glucose. You have pathways in your body where you synthesize glucose. The enzymes that are used to break down glucose, some of those enzymes can be used for the reverse reactions when we synthesize glucose. For this particular reaction, we have a lot of carbonic anhydrase in your muscle cells to produce the carbon dioxide to make carbonic acid. However, when this carbonic acid makes it to the lungs, we do the reverse reaction. There's carbonic anhydrase that can do the reverse reaction and form carbon dioxide gas, and that gas can leave via your lungs. Enzymes can catalyze the forward and reverse reaction. As mentioned earlier, enzymes are specific. They could be super specific and only interact with one particular molecule, or they could interact with a class of molecules like proteins. On this page, we show an example of an enzyme that's super specific to urea. So this is urea, and this enzyme only interacts with urea, no other molecule. Although you produce urea in your body, this particular enzyme, urease, is not found in your body. It's found in plants. Shown down here is some general information about this enzyme. It's nothing that I expect you to memorize. It works best at some optimum conditions, and we'll talk about that in a little bit. Optimum pH, optimum temperature. When we learn about those two things, we'll come back here and do some more practice problems. There's also some molecules, well, they're not molecules, they're some ions that are considered inhibitors, 2 lead cations. And I could ask a question about the lead cations and how those concentrations change. So let's stop and pause and do a problem. So here's a question. As the concentration of lead 2+ increases, the rate of the reaction would change. When you pause the video, fill in that blank. Unpause the video when you're ready to check your answer. The rate of the reaction would decrease. As you increase the inhibitor, the more inhibitor you have in there, the more likely the enzyme is not going to work as well. If it's not working as well, the rate of the reaction decreases. In a little bit, we'll come back and ask a question about optimum temperature. And maybe for fun, something about optimum pH. Some enzymes are relatively specific, meaning they react with a class of molecules with a certain type of functional group. A good example are enzymes that are called proteases. They break down peptide bonds in polypeptides. Proteases is a general name for these enzymes. There's lots of different types. I don't expect you to remember those types, but we're going to discuss them a little bit. In our example of our general enzyme action in which the enzyme reacts with the substrate, we had one in which the active site had a serine group, that alcohol group. It reacted with that amide group. And when you have an active site for a protease that has a serine residue, it's called a serine protease. If I had made you memorize all the various amino acids and you look at these various ones, besides one that I didn't highlight, something's very common with all those R groups. They have lone pairs, whether it's on a sulfur or an oxygen. And what's common for the reaction is that lone pair makes a bond with the carbon that's part of the amide group. And that's one of the steps involved in the breaking of that peptide bond. There's also active sites in proteases in which there's metal cations. And we'll see an example in which zinc the cation is part of the active site. Proteases are found in all organisms, meaning all organisms can break down polypeptides. On the next page, we'll show an example of an active site with an aspartate protease. Shown on this page is an example of an aspartic acid protease. There's lots of different types of aspartic acid proteases, and here are some names of some enzymes that are in your body. We'll see pepsin and chiromycin in just a little bit as example enzymes. But what's true about all of these is that there's a R group that has aspartic acid. So a carboxylic acid R group that's part of the catalytic process. I'm going to go through the mechanism of this catalytic process. It's kind of similar to that serine example. But note, I don't expect you to have to repeat this mechanism on the exam. It's for illustration purposes only. I'm pretty sure you didn't go over it in your organic chemistry class. But when we look at it, there's lone pairs on the atoms that are involved in making bonds. And there's lone pairs, if we can draw them, on this oxygen in that carboxylic acid anion. A lone pair is used to make a bond with this hydrogen. That bond breaks. A lone pair on that oxygen in the water makes a bond to the carbon that's part of the amide group, kind of like the serine example. When all those bonds break and made, we have an oxygen bonded to that carbon right there. And just like our electronegativity example previously, what has the stronger pull for electrons would be the oxygen. So these bonding electrons right here are kind of further away from that nitrogen, and that bond eventually breaks, which splits the polypeptide into two. And during that process, that H that's part of the aspartic acid, which wasn't there in the beginning, that H, that bond breaks, and that H becomes part of the amine group that's part of the polypeptide. So we reform the original active site structure, and we have two polypeptide chains. Kind of similar to our serine example, but a little bit different. And there's lots of different examples of mechanisms with enzymes within your body. They're not all exactly the same, and it depends, of course, on the chemical reaction. Activity, if you remember, is related to protein function. The function of an enzyme is to catalyze a reaction. So enzyme activity is related to rate of the reaction. Activity and rate of the reaction for enzymes are the same thing, meaning if the rate of the reaction is increasing, it means activity is increasing for an enzyme. So what I wrote here, for enzymes, rate of reaction and activity are the same thing. Experimentally, there's been lots of tests that have been devised to study the activity of enzymes. And these experiments are called enzyme assays. And what's been done is that you isolate an enzyme to be studied, and chemicals, reactants that will interact with that enzyme have been developed. And these are chemicals that you don't find in organisms. They're ones that have been synthesized in the lab. So yes, those enzymes react with some sort of chemical that's in your body or in plants or some organism, but what's been found experimentally are other molecules that can also be catalyzed by that particular enzyme. And the rates of reactions have been studied with these enzymes, and different variables have been altered to look at the rates of reactions. These laboratory chemicals that have been synthesized, they've also been chosen, they've been chosen to interact with the enzyme, to be catalyzed by the enzyme, but they've also been chosen for a particular chemical reaction in which the reactant either has some color or the product that's being produced has some color. That way we can use things like spectrometers, which we're going to be using in our laboratory, to look at the amount of product that's being produced, which will allow you to get rates of reactions. When the scientists developed these enzymes assays, the goal, again, was to look at how the activity changes, how the rate of those reactions change. So any variable that can change the shape of the protein, change its function, change its activity, would have an influence on rate. So all those variables that you learned about previously in the protein chapter, those variables that you can experimentally change, that alter the protein shape and conformation, would change the function, would change the activity, would change the rate of the reaction. I'm pretty sure you could list most of those. Why don't you go ahead and pause the video and list as many variables, experimental variables, that you could change that could affect protein shape. Unpause the video when you're ready to check your answer. Enzymes and shape changes can have a dramatic effect on its activity. Remember, when that substrate and the active site, when those functional groups, when they come in contact, they have to be really, really close to each other. Small little changes in the active site can have a dramatic effect on the function of the enzyme. So, what are some variables that you could change that could affect the shape of the protein? Well, one could be temperature. That affects the shape of all proteins. If the enzyme had salt bridges, pH could be a variable that you could change that could alter the shape. If there's salt bridges, the concentration of salt. If there's hydrogen bonding structures, then molecules that could hydrogen bond, if you change the concentration of those molecules, so let me keep writing, I'm going to abbreviate it as H bond, changing that concentration, changing the substrate concentration. The substrate needs to collide with the active site. A lower concentration would change the rate. So substrate concentration. There's inhibitors and activators with a lot of different enzymes. So if you change the concentration of those, that could affect enzyme activity. And these are studied. So change the concentration of an inhibitor or the concentration with an up arrow of an activator. There's other variables, but we haven't learned about those yet. So we'll come back here later after we get some more information in upcoming slides, and we'll complete it. One last note for this slide. Some enzyme assays that have been developed have been used in the medical profession. We'll talk more thoroughly about this later in our lecture notes in the very last part when we discuss medical applications, but I'm going to give a brief overview at this point. These assays that are used in the medical profession, certain enzymes that are only expressed in certain cells have been studied. And remember, all cells die and they release their components to the blood, which means these enzymes that may be specifically in the heart cell or the liver cell or some other cell eventually make it to the blood. Every cell dies, which means there's a certain concentration of these enzymes that are only found in certain very specific cells in your blood. There's a normal, healthy range for these enzymes. Lots of people that are considered normal and healthy gave blood samples, and those enzyme concentrations were studied using enzyme assays. They're not exactly measuring concentrations. What's been developed is they get the blood samples and they add some sort of substrate that's very specific for an enzyme of interest, and they measure a normal rate range. And so if you have a heart problem, the doctor would know what a normal activity range is for a very specific enzyme that's in your heart. If you thought you had a heart attack, you would go into the doctor's office, they'd take a blood sample, and they would run an enzyme assay that's specific for that heart enzyme. So they'd add a substrate that's super specific for that heart enzyme, meaning it only reacts with that heart enzyme to give some sort of rate of reaction. If the rate of reaction is in the normal range, because there should be some heart enzyme in there, then the person didn't have a heart attack. If a person had a heart attack, then there's a lot of cells that die and release those heart enzymes to the blood, and having a higher concentration of those heart enzymes means the rate of the reaction would be higher. It's a very, very, very powerful technique for measuring cell problems, cell disease in a person. And we'll give some more examples a little bit later. We'll have a laboratory, and you can see it in your calendar, called Diagnosing Disease, where we look at an enzyme that occurs that's expressed in your liver cells. On the next few slides, we're going to look more specifically at these variables and how they affect enzyme activity. The first variable we're going to look at is substrate concentration. As substrate concentration changes, the rate of the reaction changes. And as that concentration of the substrate increases, the rate of the reaction would increase, assuming those other variables that affect the conformation of the protein are constant. So we could make a plot of rate of reaction versus substrate concentration. And as you increase substrate concentration, the rate of the reaction increases. And remember, rate of reaction is also called activity. You can use either one that you want. What's interesting, at some point, there's a leveling off of the rate. And the reason that happens is your enzyme concentrations in your cells or in your blood or in your digestive tract, they're at a certain concentration. If you kept increasing more and more substrate, well, there's only so many enzymes that they can collide with. So at some point, it would start to plateau, meaning you could increase substrate concentration and not increase reaction rate. For example, you could eat a little bit of sucrose, a little bit of sugar, and there's enzymes in your digestive tract called sucrase that catalyze the breakdown of that reaction. And increased a little bit more of the sugar of that substrate, the rate would increase a little bit more. But if you had 1 pound of a candy bar with a lot of that sugar, where you have so much sugar substrate versus the enzyme, then the rate would plateau. It can only go so high. The next variable that can affect reaction rate is enzyme concentration. And just like substrate concentration, you need enzyme in there to collide with. The higher the concentration of enzyme, the more likely the substrate and the enzyme will collide and make a complex. I know I didn't write this on the previous page, but we could have. Enzyme plus substrate makes an enzyme-substrate complex held together by intermolecular forces. This rate, it's linear, so you have reaction rate versus enzyme concentration. And the rate of the reaction, it's linearly proportional, so it increases, enzyme concentration increases. And when I say concentration of enzyme, that's assuming that it's a working enzyme. And why would I emphasize working enzyme? Well, changing the enzyme concentration is a strategy. It's one of many strategies to control the rates of reactions in your body. Some enzymes are synthesized in an inactive form, and their enzymes are cleaved a little bit and they become active. A lot of proteases that are secreted into your digestive tract are made that way. There's also enzymes that can become made inactive by attaching a phosphate or removing a phosphate to make it active or inactive. So they can flip-flop between being active and inactive via some chemical reaction that occurs in your cell. It's a strategy. Changing enzyme concentration is a strategy to control reaction rates for different pathways. And we'll see that as examples a lot when we discuss more specifically our metabolic pathways in upcoming chapters. As far as I know, all enzymes are affected by temperature changes, as long as we're above that minimum temperature. And that's because enzymes, you just need a small conformational change in the active site to have a big effect on its function. Also, as far as I know, all enzymes have an optimum temperature, which means it's a temperature that it works best at. So if we looked at an experimental plot of rate versus temperature, it would look something like this. So this is a plot that shows how reaction rate changes as temperature changes. So at some lower temperature, and it's arbitrary, as you increase temperature in that direction, the reaction rate increases. And way up here, that's the highest reaction rate, that's the highest Y value. As you increase the temperature further past that maximum peak, the rate of a reaction decreases as you increase temperature. The rate at that highest peak is called the optimum temperature. This means at this temperature, that optimum temperature, the shape of that active site is the most conducive for the substrate fitting, or maybe sticking, or maybe the functional groups aligning. As you go away from that optimum temperature, there's a negative effect, there's a negative shape change on the active site. You're not quite sure what's happening. Maybe the substrate doesn't fit in as well, or maybe the functional groups on the substrate and the active site don't align as well. You're not quite sure why, but as you move away from the optimum temperature, and that means going towards higher or lower temperatures, there's a negative shape change. And the rate, the activity for the reaction would decrease. As you move towards the optimum, so if you started right here and you decrease the temperature towards the optimum, the rate would increase. That would be a positive shape change. Or what if you started at this temperature down here and you increase the temperature towards the optimum? That would be a positive shape change and the rate of the reaction would increase. And a positive change means maybe the substrate is fitting in there better, or it sticks better, or the functional groups align better. But definitely, if you have an optimum something, if you move towards the optimum, the rate's going to increase. Okay, let me read what I just wrote, starting right here. As temperature increases or decreases from the optimum temperature, the conformation changes, and it's a negative effect, meaning the activity would decrease. As you change temperature, as temperature moves towards the optimum temperature, it's a positive shape change, it's a positive effect, and the rate of the reaction, the activity, would increase. And just remember kind of our general temperature barrier. These temperature changes, you need to have a temperature change that occurs above 10 degrees Celsius to have a shape change for a protein. But even irregardless of that temperature barrier, if you move towards the optimum, the rate's going to increase. One other question about temperature. How many denaturing temperatures would there be? An enzyme's a protein. You should be able to answer that from your previous chapter. When you go ahead and pause the video and give a number. Unpause the video when you're ready to check your answer. So remember, there's one. At lower temperatures, we store biological samples at lower temperatures. Increased temperatures, it's breaking those attractive forces. So there's some sort of denaturing temperature, and it would be here. Okay, when you look at your practice problems for these enzymes, they're going to look pretty similar to some of the protein type of questions in the previous chapter. Except this time we're dealing with enzymes, which are proteins. But for the enzymes, I'll give you information about them. I'll also give you optimum temperature information and denaturing information so you can answer questions about that. As far as answering a question about optimum temperature, we could go to a previous enzyme that we had earlier. So remember this slide. Here's some information about this protein, this enzyme. Nothing you need to memorize, but I could ask a question if you were given this information. So let me read the question. So for urase, what would happen to the rate of reaction if temperature increased from 37 degrees Celsius to 55 degrees Celsius? Why don't you pause the video and give an answer? Unpause the video when you're ready to check your answer. You're moving towards the optimum temperature, so the rate of the reaction would increase. We could make a diagram. Diagrams are always nice, but the optimum temperature is 60 degrees Celsius. If you start at 37 degrees Celsius and move up to 55 degrees Celsius, so maybe right there, the rate of the reaction, the R value, is higher. The rate of the reaction increases. Moving towards the optimum, the rate always increases. Many enzymes contain salt bridges and are affected by pH changes. And just like the temperature change having an optimum temperature, a lot of enzymes have optimum pHs, which means if you made a plot of activity, rate of reaction versus pH, you could get a plot kind of like the temperature change plot. And there's some sort of optimum pH. Again, there's a lot of enzymes and there's a lot of different optimum pHs. You don't have to memorize optimum pHs for enzymes. That information would be given to you, and you'll see that when you do your practice problems where optimum pHs are given for a protein. Also given to you in those problems would be denaturing pHs. And remember, there's two denaturing pHs. There's the high pH and the low pH. At the high pH, it's the hydroxide ion that could break those peptide bonds. At lower pHs, there's a lot of hydronium ion that could break those polypeptides up into amino acids. So there's two denaturing pHs. But as far as moving from the optimum, moving towards the optimum pH, the activity would increase. It has a positive shape change. Moving away from the optimum pH would have a negative shape effect on the active site, and the activity, the rate of the reaction, would decrease. So let's go and practice that on an example protein that we gave earlier. Okay, way back to the enzyme carbonic anhydrase. The optimum pH, it's not given to you on the slide, but the optimum pH is 5. I'm going to write a statement, and there's going to be 3 blanks that you're going to fill in. When we write this problem, I want you to think about the reverse reaction. So remember, carbonic anhydrase can catalyze the forward and reverse reaction. So when we do this problem, I want you to think about carbonic acid breaking up into carbon dioxide and H2O. So keep that in mind as we answer the question. Let me read the question. As the pH decreases from 7.4, the activity of the enzyme carbonic anhydrase, it either increases or decreases, so that's the first question. And the usage of carbonic acid, so remember it's a reactant, either increases or decreases, so fill in that blank. And the production of carbon dioxide increases or decreases. So remember, this is the reaction. Look at it that way. So go ahead and pause the video. Unpause the video when you're ready to check your answer. Okay, let's go ahead and look at the answer. And I made a diagram. I did a drawing to help us understand. But rate versus pH, the optimum pH is around 5. We're starting at 7.4. We're moving, it's decreasing from 7.4, so we're moving towards the optimum, which means the enzyme becomes more active, the rate of the reaction increases. If the rate is increasing, it must mean that the reactants are being used up more, so the usage of carbonic acid increases, and the production, if the rate is increasing, we're producing more product, the production of carbon dioxide increases. This is pretty amazing if you think about it. So you have carbonic anhydrase at your lungs. As your body is producing more carbonic acid from exercise, remember carbon dioxide becomes carbonic acid, and it's the carbonic acid that flows in your bloodstream eventually to your lungs. As you produce more of that, the enzymes that help get rid of it, they work better when you're producing more. That's pretty amazing. On this slide is a table that lists a few enzymes that are in your body and their optimum pHs. I certainly don't expect you to memorize those optimum pHs. This is for illustration purposes only. Most enzymes in your body have an optimum pH close to 7, but there's quite a few that have optimum pHs that are well below that or well above it. Just because your body, because you need to live at a pH close to 7, doesn't mean that the optimum pHs for the enzymes need to be at 7. Carbonic anhydrase, its optimum pH is 5. I'm going to illustrate a couple proteases that are in your intestinal tract and in your stomach, and we'll look at their optimum pH. Trypsin is a protease that's found in your intestinal tract. Its optimum pH is close to 8. If you remember what I said about your intestinal fluid back in the protein chapter, well, I briefly said it, the pH of your intestinal fluid right after your stomach is close to 8. So that's pretty good. You have an enzyme whose job is to break down proteins in your intestinal tract, and it works best at that pH. Pepsin is a protease that's secreted in your stomach fluid, where the pH is pretty low. It can vary between 1 and 3 throughout the day. Pepsin's optimum pH is a little bit below 2, so my plot's off a little bit, but that's okay. Note that at some point, that plot ends. That's because at really, really low pHs, very, very, very high hydronium ion concentrations, there's not enough time to do an activity study because the enzyme gets broken up so quickly. One last note for an enzyme that we're going to be looking at. In one of our laboratories, we're going to be looking at alkaline phosphatase. That's the one we're going to be using in diagnosing disease. Its optimum pH is 9.5. It still works inside your cellular fluids at a pH of 7, but it works best at that pH. There's nowhere in your body where it's close to being a pH of 9.5. But when we run our assay that was developed for alkaline phosphatase, it's going to be buffered close to a pH of 9.5. Another variable that affects enzyme activity is the concentration of inhibitors and activators. On this slide, we're going to talk about inhibitors, and in particular, competitive inhibitors. In a few slides, we'll talk about allosteric inhibitors. But any inhibitor, that chemical, when it's around the protein, it can bind to the protein, and it makes the enzyme, makes that protein not function as well. Which means for an enzyme catalyzed reaction, the more inhibitor you have, the lower the rate of that reaction. All of these competitive inhibitors, they bind reversibly, meaning they bind via intermolecular forces. No covalent bonds are made. A competitive inhibitor, and here's a diagram down here to illustrate it, a competitive inhibitor, it binds to the active site. And when it's bound to the active site, it prevents the substrate from chemically reacting. It's not a permanent covalent bond, it's an attraction. Eventually that inhibitor will diffuse away. The higher the concentration of inhibitor, the more likely it'll be bound to the active site and prevent the substrate, prevent the chemical reaction from happening. So, a competitive inhibitor binds to the active site and competes with the substrate, prevents the substrate from binding, and what I wrote here, and blocks the substrate from entering the active site. And as concentration of the inhibitor increases, and we need our increase in there, up arrow, the rate of the reaction decreases. That's true with any inhibitor, whether it's competitive or allosteric. So, what makes a molecule the competitive inhibitor? Well, what is that inhibitor doing? When it functions, it has to be able to fit into that active site, and it needs to stay in that active site. The shape of that inhibitor is very, very similar to the substrate. It needs to stick in there, so there must be some sort of attraction. So competitive inhibitors, they have to be attracted to the active site, and it has to have a similar shape as the substrate. However, it doesn't chemically react, so the functional group that's on the substrate that's going to chemically react, you wouldn't see that on the inhibitor. So let's make that as a list for a competitive inhibitor. Here's a list for the competitive inhibitor. That competitive inhibitor, it has to be attracted to the active site, and it has to have a similar shape as the substrate. It needs to be able to fit into the active site. But the functional group that reacts on the substrate, you wouldn't find that on the inhibitor. It doesn't chemically react. Another note about competitive inhibitors, they bind to the active site, and the enzyme, the shape of that active site, the conformation, it doesn't change. We'll see a little bit later the allosteric inhibitor does change the shape of the active site. And there's always a chance on the exam that I may have you contrast a competitive inhibitor and an allosteric inhibitor. On the next slide, I'm going to show you an example of a competitive inhibitor. Sulfa drugs are competitive inhibitors, not in you, but in certain types of bacteria that can produce folic acid. Folic acid in certain bacteria can be produced from para-aminobenzoic acid. It's a 5-step pathway, and we're not showing the pathway, but 5 arrows, 5 reactions. But para-aminobenzoic acid, that's the substrate. The sulfa drug has an aromatic ring, just like this benzoic acid. So we'll draw that ring structure and make a little circle. It also has the amine group, so there's NH2. What's different, instead of the carboxylic acid, there's a group that I'm sure you didn't learn about in 122, but it's a sulfur-based group, and this is what makes the two different. It has a very, very similar shape, so this is our competitive inhibitor. It has a similar shape as the substrate as the reactant, so it fits in the active site of enzyme number 1, and there's a functional group on the sulfa drug just like the substrate, and this functional group is attracted to the active site. And if you look at it, and made an educated guess, it would be that amine functional group. So it's that part that sticks to the active site. It doesn't have the carboxylic acid functional group to do the first reaction with enzyme number 1. It has this group, so it blocks the substrate from entering. This sulfa drug, this competitive inhibitor, it only interacts with the active site on enzyme number 1. However, that interaction has an effect on the entire pathway. If you add the sulfa drug and you slow down reaction number 1, if you close that dam on reaction number 1, it affects the whole flow downstream of that pathway. If you shut a dam along the Spokane River, it affects the whole river flow. Slowing down reaction number 1 slows down the rate of the pathway. With that concept understood, you should be able to fill in the following statements. As the concentration of sulfa drug, if it's increased, the rate of reaction 1 would, so fill in that blank, increase or decrease. As the rate of reaction 1 increases or decreases, as you said up above, the rate of the pathway would change. So fill in that blank. And the concentration of folic acid, it's being produced in that pathway, it would change. Why don't you go ahead and pause the video and fill in those 4 blanks. Pause the video when you're ready to check your answer. So as you add more of the inhibitor, the rate of that reaction that's inhibited by it, which is reaction number 1, that rate would decrease. If you decrease one reaction in a pathway, then the pathway decreases. You produce less of the product of reaction number 1, so reaction number 2 would decrease because the concentration of that reactant decreases. Folic acid is the end product. If the pathway is decreasing, then its concentration would decrease. Sulfa drugs, for historical information, was given as a treatment to kill certain bacteria in your bladder about 100 years ago. The sulfa drug would interfere with the metabolism in the bacteria, but not in you. So how could that be? Well, you and I and all higher life forms, for the most part, we don't have the ability to synthesize vitamins. Folic acid is a vitamin. The only vitamin you and I have the ability to synthesize is vitamin D. We have to get our vitamins from our diet, which means these enzymes that are used to make folic acid, we don't have those enzymes in their active sites in our bodies. Which means if we took a sulfa drug, there's not an active site in our body that it would competitively inhibit. So you can take the sulfa drug at low concentrations and it wouldn't act as an inhibitor or an activator. And eventually, if you took it, it would be diluted and it would slowly go away. But once it made it to the area where the bacteria is at in your bladder, well, then it could diffuse into the cells of the bacteria and interfere with the production of folic acid. Folic acid, like every vitamin, is used in the metabolic pathways of that organism. And if that folic acid concentration was too low, then that could weaken the bacteria and kill it. This sulfa drug was a pretty powerful drug to kill the bacteria until those bacteria became resistant. Their enzymes mutated a little bit and the active sites changed a little bit. Another variable that can affect enzyme activity is the concentration of allosteric inhibitors and allosteric activators. We recently learned about competitive inhibitors. The allosteric mechanism, the effect on the enzyme, is quite different. For allosteric control, it can be negative or positive. Positive meaning it's an activator, negative meaning it's an inhibitor. For the competitive process, it's just an inhibitor. If we look at the diagram below, one of them is showing a positive effect and one of them is showing a negative effect. For allosteric control, the allosteric molecule, it binds to the enzyme in a different location than the active site. Remember, for a competitive inhibitor, it binds at the active site. There's a special binding site that the allosteric molecule binds to, and it's very, very, very specific to that allosteric molecule. So down here, if we look at that diagram, here's an allosteric molecule. It could be an inhibitor, it could be an activator. We'll try and figure it out. Here's the active site. So the substrate's there. When that allosteric molecule binds to that binding site, so shown there, something happens to the active site. Well, it looks like it goes away. It doesn't have to go away completely, but it changes the shape of the active site. And maybe the substrate doesn't fit as well, or maybe it doesn't stick as well, or maybe the functional groups don't align as well. But there's a shape change at the active site, and it's a negative, it has a negative effect. Which means if you had this mechanism for this diagram, it's acting as an inhibitor. In our other diagram, here's our enzyme. There's no active site, but when that allosteric molecule binds to its allosteric binding site, that active site changes shape and it makes it more conducive to the process that can catalyze the reaction, meaning the substrate can fit better, it can stick better, and the functional groups on both of them can align better. So over here, this must be an activator. Note, in our diagram, we're showing just one allosteric binding site. There's some enzymes where there's multiple binding sites for different types of molecules. And we'll see that when we give examples of inhibitors and activators a little bit later. But again, this binding site, this allosteric binding site, it's very specific to certain molecules. Just like the active site is very specific to the substrate. If you remember earlier, I said you may have to contrast on the exam the differences between an allosteric inhibitor and a competitive inhibitor. What's a little different, if we could make a list, one thing is that the allosteric controller, allosteric inhibitor, binds to a different location than the active site. And that allosteric inhibitor, it causes a shape change, a conformational change in the active site. A competitive inhibitor just sticks there and blocks the substrate from entering. Okay, let's go ahead and practice. I'm going to give you some information and you're going to answer some questions. Let's do a problem where we're first talking about an inhibitor, an allosteric inhibitor. So let me kind of draw some things to separate the statements from the questions. Let's say we have an enzyme and there's a certain concentration of the inhibitor. Remember, these inhibitors and activators, they're part of your metabolism. And those chemicals are at certain concentrations in your body. At this certain concentration, there's a reaction diagram with a certain activation energy. And let's just assume it's an exothermic reaction. So that's a certain activation energy with a certain concentration of the inhibitor. Let's assume for some reason that inhibitor concentration decreased. So there's 3 blanks that I want you to fill in. If the inhibitor concentration decreased, so a down arrow, the bond strength would change of the reactant. So we'll fill in that blank. The activation energy and the bond strength go hand in hand. So the activation energy would change. The rate of the reaction would also change. So this reaction diagram is at a certain concentration of the inhibitor. I also want you to superimpose the reaction diagram if the inhibitor's concentration is lower, and it should be based upon what you say happens to the activation energy. When you go ahead and pause the video, fill in those blanks and superimpose another reaction diagram. Unpause the video when you're ready to check your answer. If there's less inhibitor, it means the enzyme is doing a better job, and its job is to weaken the bonds. So the bond strength would decrease, so a down arrow. If the bonds are weaker, it requires less energy to break that bond, so the activation energy would be lower. And the reaction rate would be higher. So in red, if we superimposed a reaction diagram, we have the same reactant, same product, so the beginning and the end is the same, but the activation energy would be lower. So the red is at a lower concentration. Okay, let's ask a question about an activator, an allosteric activator concentration changing. So let's just say we're at some activator concentration and we have this reaction diagram for some reaction. It's exothermic and it has a certain activation energy. Let's say the activator concentration increased. The bond strength would change, the activation energy would change, and the rate of the reaction would change. Okay, so I want you to fill in these 3 blanks and also superimpose a reaction diagram at the higher concentration of the activator. Go ahead and pause the video and do those 4 things. Unpause the video when you're ready to check your answer. The activator makes the enzyme work better. If there's a higher concentration, there's a higher probability of be attached to the enzyme, so the enzyme is working better, which means the bond strengths would be lower as the activator concentration increases. The activation energy and bond strength go hand in hand. The rate of the reaction would increase because the bonds are weaker and there's more molecules that could break those bonds. As far as the reaction diagram, we're starting with the same reactants, same products, but the enzyme's working better and it has a lower activation energy, so it would be lower. So we'll just kind of draw an arrow. That's the reaction diagram with a higher concentration of the activator. Note, I could have asked a similar question, but maybe the activator's concentration decreased, and we'd have the opposite trend. Same thing with our original question for the inhibitor. I could have said the inhibitor concentration increased, and we'd have the opposite trends that we showed in our first practice problem. All of the factors on the previous page that affect enzyme activity, these are variables that your cell uses to regulate your metabolism, to regulate enzyme activity, to increase or decrease the rates of the pathways. Within the cell, so cellular control, there's definitely inhibitors and activators in you and every other organism that control the rates. There's other variables too that we're going to discuss on upcoming pages. But the concentrations of molecules that are within the cell affects the rates of all the various pathways in that cell. You're a little more complicated because you have communication, chemical communication between cells, but definitely you and every other bacteria that lives has inhibitors and activators that control the rates of reactions. These chemicals that are in your cell, these are chemicals that are part of your metabolism. Also, chemicals that aren't part of your metabolism, but they just may be within a food. There's also chemicals that are in your foods that you eat that can act as inhibitors or activators, or just environmental chemicals that you may be bringing into your body. So what I wrote, activators and inhibitors that are within your cell are part of your metabolism, and when we look at it in upcoming chemical reactions, for the various pathways we're going to look at in upcoming chapters, we'll just look at inhibitors and activators that are part of the chemical reactions that we're looking at. And these inhibitors and activators, they could also be molecules that you don't produce or use up, but they may be in your food or other chemicals that you bring into your body. Some of these chemicals that you bring into your body could have a negative effect, could be kind of bad for your pathways, or they could be a positive effect. As far as controlling a pathway contains a series of reactions like the one shown down below. In order to slow down a pathway, you only have to control one enzyme. So not every enzyme that's part of the pathway has to be controlled. But there's inhibitors and activators that control at least one enzyme for every pathway in your body. And as the concentration of these inhibitors and activators, as they change, then the rates of those pathways will change. This is extremely powerful. As the chemical environment in your cell changes, so do the pathways. And if these inhibitors and activators are part of a pathway, the rates of these pathways will change to try and keep the concentrations of those molecules that are part of the pathway constant. Or it could be other environmental chemicals you bring into your body can have a dramatic effect on the rates of pathways in your body. And remember that one broccoli example, the chemicals in broccoli that affected the 400 genes for the gene expression in the prostate cell, those chemicals that affect gene expression, which is a chemical pathway, those molecules that affect it aren't molecules that you produce, but they do act as inhibitors and activators in your pathways. A common theme in control for inhibitors and activators, a chemical that's produced downstream is usually an inhibitor upstream. So if we had a lake producing something, there's a pathway producing something else, and there could be another river from that lake, and maybe there's some molecule down here in this lake. This downstream molecule usually controls an upstream enzyme. Often an end molecule of a pathway or a series of pathways controls upstream enzymes. There could also be rivers that join each other, so this could be a key juncture point. And this is kind of showing the complexity of your pathways of your river systems. But maybe this starting molecule that's here, often this molecule controls the pathway in a river that runs parallel that feeds into the same lake. If this lake is too high, so this lake right here, it makes sense that this river should slow down so that this lake doesn't get too high. We'll see examples of that when we look at pathways more specifically a little bit later on. On this page, we're just going to look at one pathway, this particular pathway. Don't worry about juncture molecules, but this is one pathway, not a series of pathways. But what's often true in chemical pathways, an end molecule, like the isoleucine, which is an amino acid, affects its inhibitor or activator of something upstream, an enzyme that's upstream. In this particular pathway, this amino acid is used to synthesize this amino acid. There's 1, 2, 3, 4, 5 chemical reactions in a series for this pathway. The isoleucine affects enzyme number 1 and only enzyme number 1. As mentioned many times before, a strategy in your cell is to keep concentration of chemicals constant, and that's no different with these two amino acids. The isoleucine can be used to make other polypeptides. It can be used to make other nitrogen-containing molecules like choline. So we'll just write nitrogen-based molecules. It's not this amino acid is not a waste product. It can also be used, the chemical energy in that amino acid can be used to make acetyl coenzyme A, and we'll see a little bit later that can be used, in that catabolism pathway. You can also make a lot of ATP. You can metabolize amino acids to make ATP, which could be used to do a lot of other things in your body like move and build bigger molecules. The first amino acid you have to get from your diet. You don't have the ability to synthesize that. But there's a lake level here and a lake level there. They're not waste molecules. And it should make sense that the end molecule here, the isoleucine, inhibits an upstream enzyme. If this concentration gets too big, you want this pathway to slow down. That means that that enzyme should be an inhibitor. Okay, let's do a practice problem with this pathway. So I'm going to write a statement, and you're going to fill in the blanks. So here's the statement. If the concentration of isoleucine, if it increases, only enzyme number 1 is inhibited. So enzyme number 2, 3, 4, and 5, they're not controlled by that inhibitor. But the rate of reaction 1 would change. So fill in that blank. The activation energy of enzyme of reaction 1 would fill in that blank. And the rate of the pathway would change. So fill in that last blank. Pause the video. Unpause the video when you're ready to check your answer. If there's more inhibitor, that enzyme for reaction 1 wouldn't work as well, so the rate would lower, so decrease. And the activation energy, the enzyme's not doing as good of a job of weakening the bond, so the bond strength would increase, activation energy would increase. If you slow down one chemical reaction in the pathway, the whole pathway slows down, so the rate of the pathway would decrease. There's many strategies to control the pathways in your cell. If we wanted to call this one strategy, the first of many, cellular concentration of inhibitors and activators, another strategy to control rate has to do with enzyme concentration and activity. And in this strategy, in an organism, in a cell like you, the enzymes are made in an inactive form called a Zymogen. So we'll just call this number 2. And let's say that this is a polypeptide. This is an enzyme, but it's in an inactive form. And there's lots of examples of this. You don't want it to work until there's some sort of chemical signal to make it work. And a lot of those digestive enzymes that we talked about earlier in this chapter, like pepsin or trypsin, these are enzymes that catalyze the breakdown of polypeptides. You don't want them super active in the cell that they're being made. And there's no reason to constantly make it and secrete it in your digestive tract if you're not eating. So a strategy is to synthesize these enzymes in an inactive form. And often to make it active, one of the end pieces of that polypeptide here or there is cleaved off. And that's what it's trying to show here. This little line means a small piece of the polypeptide that was cleaved, and this would be the active form. In this particular example, so our example 1, this enzyme, this inactive form, this is synthesized in your pancreas. You don't need to have that memorized. And it's secreted when there's some sort of chemical signal. And it's secreted when you eat food. However, there's also another enzyme that's specifically synthesized just to break down this inactive form to make the trypsin that's active. The enterokinase is synthesized in your duodenum, and you don't need to know anatomy to take this class. And the inactive form of the enzyme, the trypsinogen, both are released when you eat food. So you get this mixture with your food, and the trypsin that's active can catalyze the breakdown of proteins, polypeptides, into the amino acid form. This strategy, as I mentioned when we first started this slide, it's basically controlling enzyme activity by changing the concentration. Yes, you have an enzyme, but you don't have a working enzyme when it's in the inactive form. This is an example for this chemical reaction, but there's lots of other examples of pathways where you synthesize enzymes in inactive form. If we had time to go over blood clotting, you have a lot of enzymes that are part of that pretty complex pathway, and they need to be made and they need to be ready, and a lot of them need to be in your blood circulating all the time in case you get a cut and you need a clot to be formed. So those enzymes, you don't want clotting to occur unless there's some sort of trigger mechanism, some chemical that can make some enzymes active to start the chemical pathways going. But those enzymes are circulating in your blood, and when you get a chemical signal, some of them become active and the pathway can start. So here's a second example of regulation of your metabolism. On the next page, we'll look at a third example. A third strategy that's used to control the rates of reactions and metabolism in your body is gene expression. So we'll just write number 3 there. And this is seen in all life forms, lower life forms like a bacteria and higher life forms like you and I. We're a little more complicated and we have a little bit more control than a bacteria, actually quite a bit more control, but still you have that same general mechanism. So remember, gene expression, that's the chemical pathway where you're going from your DNA to make RNA, and the RNAs are used to make the proteins, and proteins, well, enzymes are proteins. So if you have a higher rate of gene expression for a particular protein, you're going to produce a higher concentration of protein. So controlling enzyme activity here, again, is controlling the rate of reaction, the concentration of that particular protein, that particular enzyme. So when I was giving my broccoli example, and broccoli is really good for you, so you should eat it, but those chemicals that are in broccoli, they're controlling the different steps that are part of the pathway to make a protein. And if you remember, there's lots of different steps. On this page, it shows all the steps that we gave in the protein chapter about gene expression and protein synthesis. And remember, from DNA, the process to create messenger RNA is called transcription, And those messenger RNAs, they go into the cytosol, and that process, those steps to produce proteins, is called translation. The control of this pathway occurs at every step. Actually, I should phrase it slightly differently. A control could happen at any step. You only need to control one step of the pathway to control the concentration of the protein that's being synthesized. Let's look at the various steps in which we see examples of control in organisms like you and I. There's some genes where there actually needs to be a chemical modification of the DNA in order for it to be expressed. Most genes aren't that way, but there's a few examples of proteins in your body that are that way. This modification, any chemical process or physical process, anything that happens in your body is the result of proteins. That modification, there's enzymes that are involved, and there's control of that. There's inhibitors and activators for that. So that's one example of control of gene expression, modification of the DNA. It has to be modified so it can become active. Some genes, remember, for the gene to be expressed, the strands of the two DNA molecules have to be away from each other. Some genes, those strands are always away. Some aren't, and to get those apart, remember there's proteins that are part of that process, and there's enzymes that are part of that process. That transcriptional control to make the messenger RNA, there can be control there. The proteins that are surrounding the DNA molecules, there's sites on there where you can have inhibitors or activators. There's enzymes that are part of the synthesis of messenger RNA, and those can be controlled. So that's a second way of controlling gene expression. A third way, some messenger RNAs, even though they've been synthesized, they have to be spliced and put back together, and that's what's trying to be shown in this example. So you made a messenger RNA, and there's red portions and green portions. Well, there needs to be some splicing and putting back together to make an active messenger RNA. So that post-transcriptional control, that's a step, and that's controlled. There's enzymes that are part of that process, of those pathways, and there's inhibitors and activators. Okay, the next step, once you have the messenger RNAs, and there could be a lot of them, but you still need a chemical pathway to make the proteins. And that process is called translational. There's enzymes that are involved in that. There's lots of macromolecules that are involved in that. And there can be controlled parts to that too. Another way of controlling, I mean, we may or may not have an active protein yet, but once you make the protein chains, maybe something else needs to happen to make an active protein, like our Zymogen. Or, remember, for insulin, you have two different chains. There needs to be an enzyme that links them together, links the disulfide linkages together to make that active insulin. Those enzymes that are part of that process, they would be controlled. This process of controlling gene expression is pretty complicated in you and I. It's not nearly as complicated in something like bacteria that some of you may have learned about in microbiology, but we're a lot more complicated than bacteria. As far as knowing how every gene is controlled, we know a lot less than we do know. But we do know examples of all of these if we went to the literature. But this control can come from inhibitors and activators that are in the cell. We also have chemicals, hormones that we'll learn about a little bit later that can affect gene expression. They're called steroid hormones, and in an upcoming chapter we'll learn about chemical communication between cells and hormones. But those hormones, those steroid hormones, can go in and act as inhibitors and activators on DNA expression and affect the transcriptional process. So there's lots of examples of control of gene expression, genetic control. And this control is controlling ultimately the concentration of the protein, the enzyme that's being produced. Could make a higher concentration, increase activity, or make it at a lower concentration and decrease activity. You and the chemistry that occurs in your body and the control of that chemistry that occurs, it's truly, truly amazing. An irreversible inhibitor, this is a chemical that's around a protein, and in this example we'll use it to talk about enzymes, that forms a covalent bond with that protein and makes it so that the enzyme doesn't work anymore. This is not a strategy that your body uses. The inhibitors that we've talked about, they make intermolecular force attractions that can be made and broken. A covalent bond, you can think of it as being something that's permanent. If an irreversible inhibitor attaches to a protein or an enzyme in your metabolism, that protein's not going to work anymore. That protein's been denatured. So these irreversible inhibitors, these are not chemicals that are part of your metabolism. There's lots of examples of irreversible inhibitors, and we give lots of examples for proteins that aren't enzymes. So since we're in the enzyme chapter, we'll give an example of one that's an inhibitor, irreversible inhibitor for an enzyme. And a good example is cyanide. Cyanide is a polyatomic ion, so there's a triple bond between the carbon and the nitrogen, and there's lone pairs on the nitrogen and on the carbon. Cyanide can interfere with any protein that has a heme molecule. Remember, a heme molecule, here's an example of a heme molecule down below. We saw one like this in hemoglobin. But chemicals with lone pairs can make a bond to the iron that's part of that heme molecule. That's what oxygen does in hemoglobin. It forms a weak covalent bond with its lone pairs on the heme molecules in hemoglobin. Well, as far as an enzyme, hemoglobin's not an enzyme, but there's an enzyme called cytochrome C oxidase. This is the enzyme that oxygen reacts with in a pathway called oxidation phosphorylation. And we'll see it a little bit later. Cytochrome C oxidase is found in the mitochondria. Remember, that's an organelle found in your cells. It's a two-membrane organelle. Cytochrome C oxidase is a membrane protein. So this is trying to show the cytochrome C oxidase. There's lots of protein chains that are connected together, and all the colors show that. It's a pretty complicated membrane molecule, and it has more function than just using oxygen as a reactant. But there's heme molecules called heme A and heme C. You can kind of see that in the pictures here. The heme molecules, that's where the oxygen is going to chemically react, so they're part of the active site. And there can be a covalent bond that happens, but if the cyanide, that cyanide and its lone pairs, that cyanide can form a very, very strong bond with the iron, and if it's connected to the heme, then the oxygen isn't going to react. So cyanide poisoning, what happens is that the cyanide binds to the cytochrome C oxidase, and it prevents one of the reactions that's part of oxidation phosphorylation happening, and when that reaction stops, the whole pathway shuts down. The main purpose of that pathway in most cells is to produce ATP. And if cyanide's bonded and oxygen can't, then you stop producing ATP and the cell dies. We'll see cytochrome C oxidase. It's also called complex 4, it has different names, in upcoming pathways. You don't have to remember this example, but just know that an irreversible inhibitor, it forms a strong covalent bond, and it makes it so that enzyme's not going to work anymore. And you can think of it as being a permanent thing. It's not a strategy that your body uses. These irreversible inhibitors, these are chemicals that are coming from the environment and changing the protein, preventing it from working. And the change always involves a chemical reaction, an irreversible chemical reaction. So there you go. Irreversible inhibitors. On this page, it shows the basis for classification of enzymes. Enzymes are classified based upon the chemical reaction that they conduct. So, for example, if an enzyme is part of an oxidation reduction reaction, it falls in one classification. If it involves the transfer of functional groups from one carbon-based molecule to another, it's part of another type. Maybe water is part of the reaction, so a hydrolysis type of reaction. Okay, to be honest, I've never asked a question about enzyme classification on the exam. More than anything from this slide, I want you to know that if you see the ending of a name in biochemistry and it ends in ase, it's probably an enzyme. Most enzymes, unless they were named a long time ago, end in ase. There are a lot of practice problems in your textbook for nomenclature, enzyme classification, but you won't have one of those on the exam. On this page, we see a word that we're introduced in the first chapter called coenzymes. Coenzymes are a subclass of chemicals and biological systems that are called cofactors. Cofactors are chemicals that bind to the active site of some protein. So bind means they attach through intermolecular forces, and they give that enzyme some chemical property or structure that it wouldn't have without that cofactor. So if you remember all the coenzymes that we talked about before, like coenzyme A, FAD, NAD, those coenzymes that we mentioned in a previous chapter, coenzymes are a subclass of cofactors. So cofactors could be a coenzyme. Coenzymes, if we look at the definition down below, coenzymes are molecules. Examples of coenzymes, which are cofactors that we gave before, something like coenzyme A, FAD, NADH. These are all examples of coenzymes. They're non-protein molecules that give the enzyme some sort of chemical property. Cofactors could also be ions. So good examples of cofactors that are ions that we'll see in a little bit, something like zinc 2+, we'll see it as an example as being a cofactor. Or maybe magnesium 2+. A lot of the metal ions that go into your body are cofactors. Maybe something like copper 2+. There's lots of examples of metal ions that are cofactors. So if it's a molecule, a non-protein molecule that's a cofactor, it's called a coenzyme. If it's an ion that's a cofactor, what's kind of funny is it's just called a cofactor. Coenzymes are cofactors, but they have their own special name. Ions that are cofactors are just called cofactors. So we'll kind of make an arrow over there. So cofactors are called cofactors if they're ions. Kind of funny, but I didn't make up that definition. It's just the way it is. Certain enzymes require cofactors, and without the cofactor bound to it, it's inactive. So we could have an enzyme and say that it's inactive, so we'll just put parentheses around it, with the cofactor attached. Remember, it's not a bond, it's intermolecular forces. It can attach and make the enzyme active. So an active enzyme. Remember something like coenzyme A, it can attach to an enzyme, and there's lots of different enzymes that coenzyme A can interact with. Its job is to take 2 carbons away from a carbon-based molecule, and it'll deal with that one enzyme. But once it gets those 2 carbons attached and we form the acetyl coenzyme A, then the acetyl coenzyme A can diffuse and go to a different enzyme and give that different enzyme some chemical properties. There's not a protein that's just made up of amino acids that could do something that acetyl coenzyme A or coenzyme A could do. So there's some sort of function for those cofactors when they're attached. Sometimes the cofactors also give just structure to the active site. They're not giving chemical properties, they're just giving structure so that that substrate can fit in there. But regardless, without that cofactor attached, that enzyme wouldn't work. It's kind of without the heme molecules, hemoglobin wouldn't work. Enzymes, certain enzymes need cofactors. Not every enzyme, most enzymes don't need cofactors, but some definitely do. And we'll see examples of that when we present all of our chemistry in a little bit. And a little bit means upcoming chapters. Shown on this page are 6 coenzymes that are used in your metabolic pathways. We've seen 3 out of the 6, and the first one, NAD+, FAD, and coenzyme A. There's more than 6 coenzymes in your body, but these are just examples of 6. One common theme with a lot of different coenzymes is that we use vitamins to synthesize a variety of them. Not all of them, but some of them. And without those vitamins, and remember, we have to obtain most of our vitamins from the diet. The only vitamin that you have the ability to synthesize is vitamin D. A lot of coenzymes use vitamins that contribute to part of the structure of the coenzyme. And rates of pathways depend upon concentrations of coenzymes, which depends upon concentrations of certain vitamins. And if you knew exactly where a vitamin is used, and when I say knew where it was being used for coenzymes, and if you knew where that coenzyme is being used in your metabolic pathways, you could understand why that vitamin could have a big effect on certain functions that happen in your body if you understood the correlation to different pathways. On this slide, we're showing an example of a cofactor that's an ion. And this cofactor, it's part of the active site of a protease. And its classification of a protease, if we went back to that slide, it's a metalloprotease. That zinc 2 plus cation that's in the active site is providing some chemical property to that active site. The zinc, that cation, that positive charge, it's weakening that peptide bond that's shown here in that amide functional group. And right there is water reacting. So, how could a positive charge weaken that peptide bond? Let me draw kind of a bigger schematic. Say we had an active site and there's a positive charge there. And let's just assume that we had a bond and there's a nucleus with its proton, its nucleus with its protons, and there were some bonding electrons. And again, the strength of that bond depends upon how close those positive particles are to that average position of the bonding electrons. When this bond gets close to that active site, these bonding electrons are slightly attracted to that positive charge. And the attraction to the nucleus and that positive charge is going to move it just a little bit further away. So maybe the average position is right there. That puts it a little bit further away from the nuclei. That weakens the bond. Weakens it enough so that when the water molecule collides with the bond, that bond can break. So let's write that as a statement. So the statement that I wrote, when that polypeptide is attached to the active site, the zinc 2 plus, that positive charge, pulls the bonding electrons away from the nuclei, the two nuclei, and it weakens the peptide bonds. Lowers the activation energy, increases the rate of the reaction. That zinc that's part of the active site, it's not a permanent bond in there. It's in there through attractions. But the higher the concentration of zinc, the more likely to be bound to that active site and make that enzyme active. There's lots of examples of enzymes in your body that have metal ions that are part of the active site, giving it either shape to the active site or some chemical property. In this case, the property is that it weakens the polypeptide bond. At this point, we have to go back to a slide that we left blank. Well, we left part of it blank because we didn't know about cofactors. On this slide, we listed variables that can change enzyme activity. And now we know that cofactors, their concentrations can affect enzyme activity. So cofactors. An enzyme needs a cofactor to function, so the lower the cofactor concentration, the lower the enzyme activity. So I use arrows to make that statement. As cofactor concentration decreases, activity decreases. And vice versa if you increase the cofactor concentration. The last part of this chapter, we're going to look at medical applications of enzyme concentrations in the blood to diagnose diseases. It's used in the medical field all the time and it's extremely powerful. If you remember, we had this initial conversation back on this slide. There have been a variety of enzyme assays that have been developed to study the normal concentrations of proteins or enzymes in your blood. If you remember the description I gave earlier, when a cell dies, it releases all of its components to the blood, which includes working enzymes. Every type of cell, there's a certain rate of cell death. So there's a certain rate of putting enzymes from certain cells into the blood, which means there's a normal concentration of enzymes in your blood that have come from cell death. These enzymes have been studied. If you remember, they've been studied with chemicals that aren't part of your metabolism, but they can act as a substrate. So you choose an enzyme that's in your blood that's coming from cell death, meaning it's not a protein that's secreted into the blood to have function. It functions within the cell, but it gets into the blood because of cell death. So let's draw a picture to make this a little clearer. Say we had a cell, and it could be any cell, heart cell, liver cell, bone cell, and there's cell death, so we'll write death there. The components are released to the blood, and you have this enzyme in the blood, and your blood's a very, very complex mixture. What's been studied, you look at an enzyme that's coming from a cell that's going into your blood, you find some sort of substrate that will react with that enzyme. This substrate that you study and find is something that's not part of your metabolism. What's also done when they look at enzymes, they try and find, and they meaning the biochemist, they try and find enzymes that are specific to only certain cells, or maybe just to a specific cell. When you have gene expression, some proteins are only expressed in one type of cell, or maybe a couple cells. For example, creatine kinase, this particular enzyme, is only produced in the heart cell. It's very, very specific. So these assays, when you develop them and they're being studied, you pick an enzyme that only comes from maybe one or maybe two cells. And you pick a substrate that's not part of your metabolism that you can add that will only interact with that enzyme of interest. This assay is very specific to a cell, not to your whole body. And it can tell you if that cell has some sort of disease or not. So how do you get that information? Well, when these assays are being developed, they take samples from normal and healthy people. And they get blood samples, or it could be other biological samples. We'll just think about blood. And they take the blood, they remove the red blood cells, but they have thousands of enzymes in the blood. The enzyme that they're interested in, it's slightly different concentrations in people, but there's a normal healthy range of concentrations because there's a normal healthy rate of death for something like your heart or your liver. They add these substrates to those serum samples, and they'll get some rates of reactions, and they'll get a normal rate of reaction range. When they develop these assays, they'll develop them under strict conditions. The variables that can change in enzyme activity are held constant. So something like temperature, pH, if there's cofactors, anything that could affect enzyme activity, those conditions are established. In an upcoming lab, we're going to look at 2 serum samples, and we're going to be seeing if there's some liver damage. The enzyme that we're going to be looking at is alkaline phosphatase. And you're going to be given, for the assay that we're going to run, what's in normal and healthy reaction rate range. You're also going to be given conditions about the assay. These are things that if you ran the assay in our laboratory or in the hospital, they need to remain constant. So you're going to be given the temperature condition, the pH, concentrations of different chemicals. Note, the concentrations of the assay of, say, salts and stuff like that, it doesn't have to be cellular conditions. It doesn't have to be the conditions of your blood. That's too complex. Whatever conditions were established for the assay, that's the condition you need to run it at. Our assay, we're going to run it close to a pH of 10. Your cellular fluids, there's nothing in your body that's close to a pH of 10, but that's where the assay was developed. The only variable that should change from assay to assay, whether we run it in the laboratory or in the hospital setting, is the concentration of the enzyme. And that enzyme concentration, that's what's coming from your blood sample. Note, when we look at alkaline phosphatase, and that's the only assay we're going to be looking at, it's not super specific, meaning it's expressed in 2 cells, the liver and bone. So if we got a positive test for alkaline phosphatase, we couldn't be certain that it was liver disease. It could be bone disease, which means we'd have to run another assay that could be specific to the liver and maybe others. So maybe if you ran lactate dehydrogenase and it was also positive, then it probably, liver and liver, it's probably the liver that is damaged. Shown on this page is an actual clinical report for a relative of mine. I got permission, but this person was thought to maybe have a thyroid problem. And there were lots of tests that were run. This is just page 1 of a few pages. But some of them were looking for enzymes that were specific to a thyroid, and maybe there was a thyroid disease and an increase in thyroid death, so there would be a higher concentration of that enzyme. If you look at the measurements, some of these aren't rates of reaction, some of them are. Some of them are concentrations, but ones that have rates, they're called IU. That's a medical rate term. And if we zoom in just a little bit, all these examples down here with IU per liter, those are rates of reactions. And we do see alkaline phosphatase. There were lots of tests run. I won't get into all those specifics. But what a doctor knows, here's reference ranges. Here are normal and healthy ranges. If the patient, if their rates were above the range, so maybe above 116 for alkaline phosphatase, then it indicates that there's higher death of that cell that's producing alkaline phosphatase, which means there would be a higher concentration of that enzyme. If you fall within the normal range between 32 and 116 IU per liter, then you probably don't have death from the cell, higher rate of death from the cell that produces that particular enzyme. What's also interesting about some of these assays, they can also give a time since the injury to that cell, that tissue has occurred, and specifically for heart attacks. There's some enzymes that are super specific to the heart cell. And again, heart cells are always dying. But if you had a heart attack, then you'd have a big increase in rate of reaction. And here are some different enzymes that have been studied, and there'd be different assays, different substrates. So the x-axis, it's the time since the heart attack happened, and that's in days. And here's the activity for the various enzymes, and there's dashed lines and solid lines for the different. These are acronyms for various enzymes. But the first enzyme, the very, very solid line, that's CKMB, and don't worry about that. But it's the one that gets released the quickest from cell death, meaning it's kind of the one that's the most soluble, the easiest to get into the blood. And if it's highest in concentration, if you did all these assays for all these different enzymes, and that CKMB is highest in concentration, from time 0 all the way to this point, it's highest in concentration. So if it's highest in concentration, it's definitely been less than 1 day since the heart attack. But sometimes you've heard that these assays can tell you, they can say, well, the person had the heart attack 3 hours ago. How could they get that? Look at the ratio of the various enzymes and their rates, and you can get plus or minus an hour since the time it's been since the heart attack. So we know normal and healthy people, the ratio of the 3 enzymes since the heart attack, and if you went in for a blood sample, you thought you had a heart attack, they'd run these assays. And if you did have one, they could definitely give you the time it's been, as long as it's not too far away. At some point, those proteins that have been put into the blood, they get hydrolyzed and they go into the amino acids and reused. But if it's been less than approximately 4 days, you can get a pretty good idea since the time it's been since the heart attack. So, if you went in the hospital, you thought you had a heart attack, they would run these tests. If you fell in the normal and healthy range, then maybe it's just heartburn. These tests are extremely powerful, and they're super specific for the cells. And what's really, really nice, and you'll see it in the laboratory, you can take a blood sample. All you have to do to prepare your sample is to remove the big red blood cells. You don't have to remove any salts or any of the other different types of proteins. The substrate you're adding is super specific to your chemical. These tests are extremely powerful and extremely helpful in the medical profession. Okay, lastly, we're going to do an example problem using information from alkaline phosphatase. You don't need to memorize any of this information on this page, but on the exam, I could give you information like this and ask some similar questions. So I'm going to write down the questions, we'll write down some statements, and you're going to fill in the blanks. Okay, so here are the statements. If the concentration of formaldehyde, so that's the structure of formaldehyde, we've seen it before. If that concentration increased, which would be affected? The primary, secondary, tertiary, and or quaternary. Maybe one will be affected, maybe two, maybe all of them. You need to decide by reading the description of alkaline phosphatase. Next, what would happen to the rate of a reaction if this enzyme was working? So the rate of reaction would increase or decrease if citric acid concentration increased. I bet there's something in here about citrate. And lastly, what would happen to the rate of the reaction if the concentration of zinc, the cation, decreased? Hopefully you'll find the effect of zinc somewhere in there. When you go ahead and pause the video, unpause the video when you're ready to check your answer. Okay, so formaldehyde, it contains an oxygen. It can hydrogen bond, so it can interfere with hydrogen bonding structures. If you read through the description of this particular protein, somewhere in there, looking, the enzyme contains secondary structures. Secondary structures are hydrogen bonding, so that's a yes to secondary. Note, I skipped primary. Something that can hydrogen bond can't break peptide bonds. So no to primary, yes to secondary. Okay, what about tertiary? Are there tertiary structures? So if we look in there, keep looking. Oh yeah, it contains all 4 tertiary interactions, and one of the 4 is hydrogen bonding. So that's a yes to tertiary. Okay, quaternary. Is alkaline phosphatase, is there only one chain or is there multiple chains? Well, it says it's a dimer, so that means it's 2 chains. And hopefully it tells us how those 2 chains are held together. And they're held together by hydrophobic. That's a no. But yes, there's hydrogen bonding interactions, so quaternary structures would be disrupted too. So formaldehyde, for this particular enzyme, this particular protein, would affect 3 out of the 4 structures that give rise to the protein, which give rise to its function. I don't think alkaline phosphatase would work too well in a high concentration of formaldehyde. Okay, next in blue, what happens to the rate of the reaction if citrate changes? Citrate. Oh, there's citrate. Work backwards. It's an inhibitor. The inhibitor concentration is increasing, which means the enzyme wouldn't work as well at higher citrate concentrations, so the rate of the reaction would decrease. So citrate, it's an inhibitor. The rate would decrease as the inhibitor concentration increases. Okay, zinc 2+, where's zinc at, and what is zinc? There's zinc. It's a cofactor. And you can kind of think of cofactors, they act like activators, except they're not activators. But without that cofactor, the enzyme doesn't work. The less cofactor in there, the less likely it's part of the active site, and the enzyme doesn't work without that cofactor. So as that cofactor concentration decreases, the rate of the reaction would decrease. Has the same net effect as an activator. But of course, it's not an activator. Activators are listed, and we didn't ask any questions about those.